Hello, good afternoon, students. Welcome back to Botany second year class. So now, today we have already finished the LAQs of two chapters. That is, three LAQs out of this. One LAQ will be coming, and the SAQs from the 13th chapter we have finished, and the 12th chapter we have finished. So maximum we will get from the 12th chapter one SAQ, and the SAQ from the 13th or 14th we will see, because 14th chapter we need to go. So we will complete that 14th chapter also. And meanwhile, the most important chapter where one SAQ and one VSAQ is going to be coming, that is the 9th chapter for us, that is the genetics part. So in this, once we know about the genetics and how to write the answer and how to represent the representation of the crossing, that is the one what we have to take care over here. And if you follow a small logic over there, then you won't get confused with the crossings which we are representing. And the representation only very, very important over here, how we are going with the answer. Okay, so here SAQ starts with the first SAQ is why Mendel selected pea plant for his experiment. Okay, why Mendel selected pea plant for his experiment. So here we can say the points directly. Okay. Mendel selected pea plant for his experiment. Okay, Mendel selected only pea plant. We can say it as only pea plant for his experiment. Why he has selected only pea plant? 6 points are there in this. What you have to write? The first one is plant is a bisexual plant. Okay, when the bisexual plant is there, there is no need for the hybridization to go and search another plant acts as another parent. So it's easy for us to go with the hybridization process now. So that's the one what he's get through this. There is a plant is a bisexual plant. And second one, the plant can go with the cross-pollination. This is the most important one in the pea plant. Pollination occurs by piston mechanism. We know this. Okay, wing petals, standard petal, the same one what we learned in the first year. So here also cross-pollination is the one what we can see. Apart from the cross-pollination, the self-pollination also occurs in the pea plant. So for this sake only, he has selected the pea plant for his experiments. The plant is a bisexual plant with the bisexual flowers. Bisexual flowers. Bisexual plant means bisexual flowers are present, and cross-pollination can be seen in the plant as well as self-pollination also possible is there, possibilities are there. So cross-pollination can be seen. If not, self-pollination can be done in the plant body. And the third point, offsprings are more in number. Offsprings are more in number. Because of this only, he has selected the pea plant. Offsprings means seeds. Seeds are more. Whenever the seeds are more, then we can grow them in a large number for the next generation to see the hybridization point. That is the whether the selected plant characters are seen in that plant body or not. Otherwise, those are coming as it is in the next generation or not. That's the one what Mendel thought to take out the plant. So this is the third point. Now comes the fourth point. What could be the fourth one? The flower is having the contrasting characters, specific contrasting characters in them. Contrasting characters means same character which is having the two opposite natures. That is the contrasting characters. Once you wear a dress, okay, that is the pant shirt, otherwise the top and the bottom. So if both are same color, then the other things should be different one, isn't it? If you are wearing a pant shirt, if both are having the same color, then you wear a blazer with the different color. Otherwise, if you want to wear the blazer and the pant of the same color, then the shirt will be in a different color. So the glow will be coming for the shirt as well as for the blazer. So like that in the female also, top and bottom. If both are having the same color, then the dupatta will be in a different color. So that will be the attractive one, isn't it? So that's the contrasting character. So the plant is having the contrasting characters. Okay, specific contrasting characters and more contrasting characters when we compare with the other plants in the pea plant only. 7 different contrasting characters he has identified. That means 14 characters he observed in the plant body. So that's the most important one in one single plant body only. Like that, we won't see in any other plant these many contrasting characters. So because of that, that reason only, he has selected the pea plant for his experiments. And the flowers are very big in size so that he can do the pollination very easily. Okay, and the specific colors, contrasting characters, and the flowers are big. Not that much big, but we can say flowers are big. So like this, he has taken out the contrasting and he has taken out the different characters why he has selected the pea plant, and he listed out all these points also in his book. Okay, so what they will give is this one, that is the first question. Now comes the second question. So before going to this second question, that is the differences between dominant gene and the recessive gene, and the homozygous and the heterozygous. You need to know about the descriptive part of this. That means what is meant by dominant and what is meant by homozygous and the heterozygous. Whenever a character is represented in an organism, according to Mendel, in this chapter wise, because what now I am saying is having the 3 questions answers. That means the just background work, what I am going to explain right now. Any character is represented when each and every character is having the 2 contrasting alleles for this. That is the representation of the character can be done with 2 alleles. If the 2 alleles are same letter, then that is called as the homozygous. And if the 2 alleles are present in a different font, different manner. That is the contrasting alleles. Whenever we have seen, one letter is the capital one and another one is the small, that is called as the heterozygous nature. Heterozygous nature. And in this, always the dominant character always represented with the capital letter. If tallness is the dominant character in the plant body, then it should be represented with a capital T. And opposite to this, contrasting characters for the tall is dwarf. Very short plant, that is a dwarf, what we'll say. This character should be represented with small letter. Okay. And if you want to write the T like this also, right. But here, don't forget to put this horizontal line over here. If you are forgetting, then it looks like L. If you show it like this, then it cannot be the small t. This is a blunder, and we will say they're not at all represented in a right manner, and we can have the full rights to cut the marks over here. So mind it. Whenever you are representing, representation should be in a proper way. For that sake only, might be you lost the marks. Okay, it should be very clear. So what I will say, I will suggest you right now is represent this with the, in this manner, small t. Then there is no confusion, because whenever we are writing like this stroke, we won't leave it without putting the horizontal line. Make sure it, so that it represents the small t. Then it's not the small letter, capital letter means one is very big, one is small. This is not the capital and the small letter. Case sensitive. Okay. Take care of all these things. This is the dominant character always represented with the capital letter, and the recessive character is always represented with the small letter. Okay, like this. And when the dominant and the recessive alleles are present in representing the character for the plant body, then that is called as the heterozygous in nature. Because here always the dominant characters will be expressive, recessive character will not be expressive. So here two different types of alleles are there. That means two different types of genes are present. So that is called as the heterozygous in nature. And when the character is expressed with only one type of genes, that is the capital T, or capital T always represents tallness. And if small t, then it represents dwarf character. Okay, dwarfness in the organism. So like this, we have to represent this part, and we should know what is meant by homozygous and what is meant by dominant. Always the dominant characters will be expressive. In the heterozygous condition, only the dominant characters will be expressive and the recessive characters will be expressive when both the alleles are of same kind. That means when a dominant gene is there, then the recessive gene is also there in an organism, in the allelic representation, then it won't be expressing the recessive characters. It expresses always the dominant characters. That's what we are saying. The character is dominant over this. That means this character is going to be suppressed. Okay, but it is present in the organism, but it cannot express its characters. The gene is present, but it cannot express its characters. So here always dominant characters only expressive, recessive characters will not be expressive. When the recessive characters will be expressive, when the allelic representation is the homozygous. That is, both the alleles are of the same small letter. That is the recessive representation. Then only the recessive characters can be expressive in the organism, whatever may be the organism. The organism may be the plant, animal, otherwise the human being, any one. Only this is the most important one. Even we will see in us, in the parents, if one is having the commanding power, there is the talkative, otherwise anything, then that is not coming in all the offspring. That means in all the children, only few. Then what will say, that is the dominating. If all the ones are having, if 3-4 kids are there for that couple, then 4 are having that character, then we can say that's completely the dominant character than the other parental characters. So that's the one what we are saying. Now we can go with the 3 questions at a stretch. If you know this single point, then we can go very easily with the 3 questions answers. The basic is the most important one over here, where you people were lacking. Okay. So here the second question, the differences between the dominant and the recessive. So what we learn now, what we have to say is now very clear now. Dominant characters. That means always it is a dominant. Always the allele is dominant over recessive allele. Recessive allele. Okay. And this, for the recessive allele, what we can say? Recessive allele is not dominant over the dominant allele. And this is present only in the homozygous. Recessive allele cannot be dominant cannot be dominant. That much we can say for the dominant and the recessive, the first point. Now comes the second point in this. What could be the second one? Dominant characters are represented with the capital letters and the recessive characters are represented with the small letters. Okay. Second point is represented with capital letters. And this is represented with small letters. Okay. Now, the third one. In the homozygous or in the heterozygous condition, dominant characters will be expressive, isn't it? It is expressive in homozygous or in heterozygous condition. Just now we have seen now. Same thing we are writing in the sentences. That's it. Now, in the recessive one, what we can say? It is expressive only in homozygous condition, isn't it? Expressive only in homozygous condition. Example you have to write. Give the example for this. What is the example in this? Capital T for the dominant, for the homozygous, and the heterozygous, capital T, small t. So easy for us to represent this. Example, capital T, tallness, and capital T, small t also giving the tallness only. Whereas in the recessive one, example, what we can say, dwarf character is represented with small t. But we cannot give any other representation for the dwarf because it's always expressive only in the homozygous condition. That's it. So with this, we can easily conclude the answer, isn't it? Now comes the second one. That is the homozygous and the heterozygous differences. What is meant by homozygous? Homozygous and heterozygous. What is meant by homozygous? When the allelic in the allelic representation, when both the alleles are of the same kind, that is called as the homozygous. Capital T. And one more example, small t. This is the homozygous representation. Okay, heterozygous. When the alleles are of two different kinds, what we are taking for representing the character, that is the heterozygous condition. That is, example, capital T, small t. That is one dominant allele and one recessive allele. Then only we can call it as an heterozygous condition. Okay. Now in the homozygous, whenever the alleles are in the homozygous with the small case, with the recessive characters, they will be expressive always. Only the recessive characters will be expressed in the homozygous condition. Okay. And the dominant characters will be expressed in the homozygous as well as in the heterozygous condition. The same point what we have written for the dominant and the recessive, same one we are writing in a different way over here. Okay. And the next point, what here we can say, in the homozygous condition, neither the allele is dominant over the another. Okay. Otherwise, no allele is dominant over the another allele. Both the alleles are representing only one character in the homozygous condition. Whereas in the heterozygous condition, only the dominant characters can be expressive. Only dominant characters will be expressive. This is the most important one, what we are saying and what we are giving the representation. Okay. Now comes with the most important question, how we are writing in the exam point of view as gives much more impact. Okay. So somewhat interesting, and what you learned till now, I don't know, and how you are going to give the answer also, I don't know. But like this way you have to give. That's what I'm going to say right now. Describe the test cross. The question is, define and describe the test cross. Simple. But in this, how we have to answer the question is the one, the skills can be expressed over there. Okay. And what you, the different materials, what I have seen till now, they are giving the answer in 1 or 2 lines, and they are not express, that is not elaborating the expression point of view also. So the expressive way need to be elaborated. That means need to be explained with the theory. Then only the representation and the theory will match. Then only the answer gives the worth over there. Without the explanation, if you are representing the answer, it does not have the full worth. Okay, worth should be given. So here, test cross. What is meant by test cross? The first sentence should be the definition for the test cross. When the F1 individual, I'm not writing here completely, but I am saying 2 times and say the definition. When F1 individual crossed with one of its parents. Okay, that one of its parents is always recessive parent. So F1, when the F1 offsprings crossed with recessive parent, that is called as test cross. So why we are going to explain this test cross? That is the second point what we have to say here. So in the second point, what we can say, to check the heterozygosity of the F1 generation, to check the heterozygosity of F1 generation, we will do the test cross. So for explaining the test cross, we need to take the monohybrid and the dihybrid F1 individuals. And here the explanation should be there. So while giving the representation, I am explaining the points also what we have to say. Okay. When we are saying the F1 individual, we cannot write the F1 individual at once. We have to say the original parents. Okay, original parents, because we are saying in the definition that F1 individual crossed with the recessive parent. Where are the parents? If you are saying only the F1 individuals, then how can you say the recessive parent is of this kind? Isn't it? So here we have to take the parents, the plants. So here follow the rules and regulations. In the genetics, always you have to give the labeling, that is the representation of the parents, offsprings, and the generations and the gametes always on the right side, just like the labeling part for our subject wise. Okay, whenever we are drawing the diagram, where we will give the labeling part, always on the right side now. This is the rules for the botany and zoology. Like that, here also for genetics, always the labeling part should be on the right side, not on the left side. Because in the reader and in the materials, they have given on the left side. Left side always will be having a margin. There, we should not represent anything. Okay, only on the right side we need to write. So in the, when we are taking the monohybrid, that is the, when the tall plant is crossed with dwarf plant. So tall plant always the dominant character represented with a capital T is crossed with dwarf plant small t. These are the parents. Now, the gametes which are coming from the homozygous. One single gamete will be coming. So, these are the gametes. Okay, once we cross these two, then we are getting the F1 generation with capital T, small t. So here, what is the phenotype of this? Because always the dominant allele will be expressive in the homozygous or in the heterozygous. So here the phenotype is tall. Right, the tall over there. Now, what we have to do in the F1, in the test cross, F1 has to be crossed with its recessive parent, isn't it? So, what is the F1 plant? Capital T, small t. What we have seen over here? What we got? Capital T, small t, crossed with its recessive parent. That is small t. What we are having already here? Recessive parent, isn't it? Dwarf plant. Same thing we need to represent here. Now, the alleles in the homozygous condition, the alleles when they are present in the homozygous condition, only one gamete is going to be formed in the haploid condition. So, from the small t, that is from the recessive parent, we are getting only one gamete, that is a small t. And always gametes, that is the haploid condition of the allelic representation, needs to be encircled. That means we have to cover that with the circle, protected. Otherwise, we can say with this representation that this is haploid in nature. That's the one what we are showing. Because when two haploid gametes are fused, then only we'll get the diploid offspring. Same thing over here. So, need to be represented. Okay. And from the heterozygous condition, from the parents, we will get two gametes, capital T and the small t. Okay. So now the probability you have to check it out over here. This will combine with this. Otherwise, this may combine with this. So I'm writing that in the next one. Capital T, small t. When crossed with a small t, what here we will get two gametes, na? One is a capital T, another one is a small t. And here only small t we will get. So these are the gametes. And these are parents. That also second parents in the. So first parents and second parents need to be mentioned. The rules need to be followed. Otherwise, that's a blunder, whatever we are saying. These are the first parents. Okay. Parents one, or simply we can say parents. When we are using the offspring of that generation, then those offsprings are considered as the parents. Now these are considered as the second parents, parents two. And the gametes need to be represented, gametes. Now the probability what we will get, see. Here we may get capital T, small t of one offspring, otherwise small t of this kind also we can get. That means, what is the phenotype of this? That is the tall and the dwarf, isn't it? because capital T, small t always expresses the dominant character. So these plants are always tall in nature, and the small t always represents the recessive character, that is the dwarfness of the plant body. So these are the dwarf. So here what we have to say, the phenotype of the generation. So these two plants are now given as the F2 generation. Okay, so in the F2 generation, we got two offsprings. In these two offsprings, now we will see the phenotype and the genotype. Phenotype is tall and the dwarf. So here we cannot say comma over here. We have to say it as tall is to dwarf. Okay, now the phenotypic ratio. Phenotype. Tall is to dwarf. Now the phenotypic ratio. Phenotypic ratio. How many tall plants we got over there? Ratio means number of plants what we got with this F2 generation, in the F2 generation with the test crossing. So here, how many tall plants? Only one tall plant and one dwarf plant. So here we have to give the numbering of these organisms. 1:1, isn't it? And the genotype. Genotype. Genotype of the tall plant is capital T, small t, and the dwarf plant is small t, small t. Isn't it? And now comes the genotypic ratio. Means how many plants of this kind we got in the F2 generation. That also 1:1 ratio. Okay, this is for the monohybrid cross. So here what we can say, when in the F2 generation, when the recessive characters were seen, then the F1 hybrid plant is always heterozygous in nature. Cannot be the homozygous in nature. That's the conclusion what we have to write. So once we have seen with the dihybrid cross, the test cross, then in the dihybrid cross, what will take? Yellow colored for the flower, for the fruit, and the round seeded nature. Yellow round character. Capital Y, capital Y, capital R is crossed with yellow to the contrasting character, that is the recessive character for the yellow is green, and for the round it's the wrinkled. So here the representation should be small y, small r. These are the first parents. Okay, now how the gametes are coming over here? Capital Y, one capital R. That should be encircled, and here small y and small r. That should be encircled. So that is called as the gametes now. So these two gametes will fuse together to form capital Y, capital R, small r. So this is the F1 generation. Okay. So in the F1 generation, what is the phenotype of these plants? Those are the yellow round seeded. Okay. Yellow round seeded, that is yellow color and the round seed for the round shape for the seed. So this is the F1 generation. Now, what is the test cross? F1 need to be crossed with one of its homozygous recessive parent. So capital Y, small y, capital R, small r should be crossed with small y, small y, small r, small r. These are the second parents. Parents 2. Now comes the gametes. So in the dihybrid, what we have to take the help of the Punnett square picture. Four gametes we will get. So here we should have 4. In this, one is male, one is female. So in this, what we have to give here is the heterozygous gametes. Capital Y, capital R, capital Y, small r, small y, capital R, small y, small r. And from this parent, we will get from the recessive parent always small y and small r. Okay. Now comes here how the arrangement. That is the probability. Capital Y, capital R, small r. That is the 1. And capital Y, small r, small r. And here small y, capital R, small r, small y, small r, small r. Like this, we will get the 4 different types of offsprings. So this is the F2 generation, what we will say. And these are called as the gametes. Okay. Now, what is the color? This is the yellow round. So I am giving the 4 offsprings, 4 colors. This is orange. This color is also not visible. So we will go with the yellow wrinkled. Okay. Now small y always represents the recessive character. That is the green color. So this is the green round. Capital R is there now. So it's representing the dominant color. Okay. Dominant shape. That is a green round. And here it's the green wrinkled. Like that, we will get 4 different offsprings with the 4 different characters in them. There is a yellow round, yellow wrinkled, green round, green wrinkled. So what is the phenotype of this now? Phenotype is 4 different offsprings now. That is, the first one is the yellow round. And the second one is yellow wrinkled. And the third one is green round. And green wrinkled. Like this, four phenotypes can be seen in the F2 generation. In that, what is the phenotypic ratio? How many plants of this kind we have seen over here? There is a phenotypic ratio. Phenotypic ratio is always 1:1:1:1. That means in all the cases, we have seen only one plant was there with this different phenotypes. So 1:1:1:1. Now, genotype of this. When we are saying the genotype, again, 4 genotypes will be coming along with the 4 different phenotypic characters. Now, what is the genotype? Capital Y, small y, capital R, small r. That is the yellow round, heterozygous, complete heterozygous. See here, capital Y, capital R, small r. And next comes the yellow wrinkled. Yellow wrinkled, capital Y, small y, wrinkled na, small r, recessive condition. Now comes the green round. Green is a recessive character. Small y, round is the dominant, capital R, small r. 2 dominant letters are not there for the round character. That means 2 capital R's are not there now. So 1 is also sufficient to express the character. So here 1 is the capital letter and another one is the small, that is a recessive character. And now comes the green wrinkle. Green wrinkle is the completely recessive parent what we are having already. That is a small y, small r, small r. So what is the genotypic ratio of these plants? Genotypic ratio means how many plants of with this genotypic nature we got in the F2 generation. That is also 1:1:1:1. So like this, we can conclude our answer. Okay, so see here, the phenotypic and the genotypic ratios are same in the test cross and even in the monohybrid cross. Genotypic and the phenotypic ratios are same. That is the conclusion for us, what we are going to write over here. Okay, so what we have to give the conclusion in the test cross is meant to test the heterozygosity of the F1 generation. So here the F1 generation, F1 plants should be heterozygous. Why we came to this conclusion? In the offsprings, when we have seen the recessive plants, in the offsprings, we have seen the recessive plants now. I'll show again this. See, in this, one is the completely recessive plant we are seeing with the small y, small r, small r. Like, that means the parent of the F1 generation, that is the F1 hybrid, should have the recessive gene in it. Then only this recessive character will be coming in the F2 generation for us. So like that, what we can conclude right now? The offsprings should, the F1 offspring should have the recessive gene in it. So then only in the F2 generation, we will get the one of the offspring with the completely homozygous recessive characters. So with this, we can say the F1, the test cross is meant to test the heterozygosity of the F1 generation. And in this one, what is the another conclusion over here? The genotypic and the phenotypic ratios are same. 1:1:1:1 in the dihybrid cross and 1:1 in the monohybrid cross. So the same one what we have to write in the conclusion. Then the answer looks very beautiful and you will get 4 out of 4. Without this, if you are giving only the definition for the test cross and writing the why we are doing the test cross and for namesake when you are giving the cross, that also don't, when we are starting only from this. So where is the recessive parent? So everyone will get it out. Even I am correcting this, you are saying that the test cross means the cross between the F1 generation, F1 hybrid to the one of its recessive parent. Then how the hybrid came and where are the original parents and where is the recessive parent. That means original parents need to be represented and need to be shown in the cross. So this is mandatory. Without this, without the original parents, F1 cannot came into the limelight. So if you are not showing means the F1 does not came from the parents. So parents need to be represented first. So this is the representation what you need to do. And you may get a doubt. In the reader, it's not there. Because in the reader, it's a continuation with the Mendel's laws. No need to say there that every time whenever they are saying the test cross, otherwise any other cross, they need to mention the previous one. Never. When we are giving the answer, how you are saying the answer is the technique. Your skills need to be highlighted over there, not the textbook skills. So find this and follow this and do the best work. Don't give the namesake work where you will get namesake marks. And you feel that I have written the answer. It's not that. When you are writing only the definition and the representing not with the original parents, it's not worthful for giving the 2.5 marks also. Sorry to say. Not only 1 mark or 1.5 mark we will give if you are representing only from the F1 generation. Remember this point. So from here, when you start, we won't give because you have not mentioned about the original parents. And the original parents were not mentioned. F1, it's not came from the A. So need to be represented. Always the rules need to be followed. Without a key, you cannot start any vehicle. That means don't say that now electric vehicles are coming only the button. Button is also a key, na? Okay. So if you are saying like that, then I can say many things. So don't go in that way. Everything should have a key. That is the starting point. Without the button or without the key, can you start a vehicle? You are not a thief to put the wires. Then also there should be a joining. That means a key only, na? The start is a key. So that is the one. Here also without this, if you are writing that, you are stealing the answer. That means it's not worthful. So need to be mentioned like this. So what you have to write here, what is meant by test cross, then what we have to write, how the crossing can be done. All these things need to be written in a proper way and the conclusion is mandatory. That means by seeing the recessive offsprings in the F2 generation, we can come to the conclusion that the F1 generation is always heterozygous in nature because the recessive characters are going to be expressed only in homozygous condition, not in heterozygous condition. And the F2 generation, in the F2 generation, the phenotypic and the genotypic ratios are same. Okay, so with this, we can conclude for today's class and we will go with the other important questions from this chapter. That is the law of dominance is there. And next comes the codominance and the incomplete dominance, and the mutations are there. That means 4 questions. Okay. So for how to write these answers is the most important ones. So we will discuss those answers in the next class. Stay tuned to the same to the second year botany class from 1 o'clock 1 p.m. So don't forget to subscribe our channel also, right? Thank you.